Hello and welcome to today's webinar. Integrated fragment-based approach reveals enzymatic inhibitors with potential therapeutic application. On behalf of Drug Target Review and Nanotemper Technologies, I'd like to thank you all for attending. I'm your moderator, Ikra Farouk. Today's speakers will be Antonio Masciarulo, Associate Professor at the Department of Pharmaceutical Sciences at the University of Perugia, and Alice Coletti, Postdoctoral Fellow at the Department of Pharmaceutical Sciences at the University of Perugia. Following their presentations, we'll move on to a live question and answer session where you can pose questions to today's speakers. We encourage you to submit questions at any point during the webinar using the Ask a Question panel situated below the player. So without further ado, here are today's speakers, Antonio Masciarulo and Alicia Coletti.
So thank you for the nice introduction. In the webinar of today, we are going to talk about how we have used integrated fragment-based approach to disclose novel enzymatic inhibitors with potential therapeutic applications. In the first part, I will give you an overview of our target. Then Alice Coletti, my postdoc, will explain briefly how we have been able to set up a screening cascade to identify these inhibitors, and then I will conclude with some current works that we are doing in this field of research. Our target is Indoleamine dioxygenase 1. These enzymes catalyze the first and rate-limiting step of the kynurenine pathways in which many bioactive metabolites are produced. In particular, these enzymes share 43% of sequence identity with another isoform, IDO2, albeit the second enzyme, IDO2, is not able as IDO1 in catalyzing this reaction. The structure of the enzyme is composed of a large lobe at the bottom and a small lobe up to its catalytic site in which it anchors the heme cofactors. At the level of the small domain, two immunotherapeutic receptor inhibitory-based motifs are present that are associated with specific function of the enzyme. Interestingly, the small domain of IDO1 is not concerned with its ancestral enzyme TDO, albeit both enzymes share the same catalytic pocket and the same catalytic reaction. From an historical point of view, the discovery of IDO occurred in 1957. Then, in the 80s, it was recognized that IDO is induced by interferon gamma and involved in the host response to microbial changes. Then, in 1998, there was the breakthrough discovery that IDO is connected to the immune tolerance, and its activity is essential for protecting the fetus from maternal immune system. Then, at the beginning of 2000s, it was discovered that IDO1 was involved in tumor immunoediting, both as component of the tolerogenic mechanism expressed by dendritic cell that cross-present tumor antigen, and as component of the tolerogenic mechanism expressed by tumor cells that directly suppress immune effector T cells. In particular, in cancer cells, we have an upregulation of IDO1. The same occur in dendritic cells in the tumor microenvironment. As a consequence of these increased expectations, we have actually a depletion of L-Tryptophan and increase of L-kynurenine. Then L-kynurenine promotes in T cells activation and proliferation of T regulatory cells and T cell anergy, apoptosis, and inhibition of proliferation. On the basis of this evidence, it has been proposed that the enzyme may indeed constitute a novel therapeutic target for the development of cancer drugs. And in this framework, a large amount of medicinal chemistry effort have been devoted to design and develop inhibitors of the enzyme. We may have both competitive inhibitors from designing analogs of the substrate, natural product, non-competitive inhibitors, and even more recently, people have discovered compounds that are able to bind an apoform of the enzyme. That means an enzyme without the cofactor. If we see in these slides, there was from the beginning of this century till today, a large amount of inhibitors that have been disclosed in literature and in patent applications. Some of these compounds have successfully entered in clinical stage, and in particular, the most advanced candidate today is Epacadostat. Now, unfortunately, very recently, it has been reported that these compounds failed in phase 3 clinical trials to produce statistical significant results when used in combination therapy with other immunocheckpoint inhibitors, such as Keytruda, to treat melanoma. This has increased the awareness in the scientific community that IDO targeting strategy may suffer from shortcoming in the knowledge of the complex IDO1 biology. In particular, some IDO inhibitors have indeed been recently reported to show activity at the aryl hydrocarbon receptors. Briefly, these receptors is able to upregulate IDO1 gene expressions. As a result, the enzymes increase its catalytic activity to the production of L-kynurenine that is indeed an agonist of these receptors. Now, when we have inhibitors that bind IDO1, on the one side, these compounds may inhibit the catalytic activity of IDO1 and reduce the level of kynurenine. But if these compounds are also able to bind and activate AHR, these receptors may increase the levels of IDO1 and then bypass the catalytic inhibition of the compounds through the enhancing of the expression of IDO1 gene. Another important aspect that actually constitutes a black box of the enzymes is that IDO1 is a moonlight protein, in the sense that besides having catalytic activity, this enzyme seems to have also signaling activity. In particular, this signaling activity is associated to the eating motifs that are localized at the level of the small domain. Upon phosphorylations, these motifs are able to recruit protein binding partners, including SOX3 and SHIP phosphatases. Upon the interaction with SOX3, we have the reduction of redox-1 half-life and inflammatory responses. Upon the interaction with SHIP phosphatases, we have the prolongation of IDO1's half-life and then long-term immunotolerance. Now, the aim of our work was to further investigate the moonlight function of IDO1. And in particular, we wondered whether small molecules could exist that can diversely modulate the catalytic activity and the signaling activity of IDO1. And in particular, we tried to address this query exploiting an integrated fragment-based approach to discover next-generation IDO1 ligands. Now I will provide a brief introduction on the fragment-based approach. In particular, the fragment-based paradigm in drug discovery was introduced in 1999 by researchers working in pharma companies who analyzed properties of successful drugs and hit or lead compounds from which they were developed. As a result, researchers found that some key properties, in particular molecular weight, significantly changed, increasing its average value with respect to other properties. Now, on the basis of this observation, they claim that there is a higher chance to develop drugs when starting from fragment-like compounds during hit-to-lead and lead optimization cycles, whilst a major challenge may still exist when starting from compounds with higher molecular weight. On this basis, researchers introduced the concept of rule of three to identify fragments in the molecular space with molecular weight below 300 Dalton, hydrogen bond donor below 3, hydrogen bond acceptor below 3, torsional angle below 3, and C-log P below 3, and then use the region populated by these compounds as base camp to start navigations toward the biological relevant region of the chemical space. Now, it is worth noting that in the case of our target, the 43% of IDO inhibitors reported in literature and patent applications has already molecular weight below 300 Dalton. Hence, starting our discovery program with fragments, we basically avoided the risk to leave out biological relevant area of the chemical space populated by these very small compounds that still could represent interesting hit candidates to develop novel inhibitors of the enzyme. So in summary, the advantages of using fragment-based paradigm in drug discovery is that we have an high efficiency in sampling biological relevant regions of the chemical space using thousands of fragments rather than millions of small molecules with expected higher hit rates. Fragments have also low molecular weight and are more polar and soluble than large drug-like compounds, translating into molecules with favorable pharmacokinetics, bioavailability, and toxicological properties. Moreover, biologically relevant regions of the chemical space are populated by known IDO inhibitors that have fragment-like properties, and so using this approach, we can also include in our screening campaign this part of the chemical space. As disadvantages, fragments, however, requires biophysical, biostructural screening approach since they bind with very low affinity to a protein target, so we need a very sensitive method to detect these binding activities. This may in part provide an explanation to what we have at the bottom of these slides. In a very recent work published actually the last month, we can see that only the 5% of drug candidates published in the Journal of Medicinal Chemistry derive from fragments. So it is possible that in the near future, these percentages will increase as it will be more widely used biophysical and biostructural screening approach. So the workflow that we use is composed actually of 4 parts. In the first part, we run a virtual fragment-based screening campaign to identify small molecules in libraries of commercial available molecules. Then we use biophysical screening to identify true binders of the enzyme. Then we use cellular assays to assess both the division activity in cell and biochemical assays. And then we use a preliminary pharmacological assay to assess also the signaling activity modulated by the compounds that we identify. I will give now the headphone to Alice that will explain you these steps.
Hello, everybody. Okay. So, in the first step of our workflow, we screened a library of commercially available compounds to identify molecules with fragment-like features, such as molecular weight lower than 300 Dalton and C-log P lower than 3. We filtered these molecules by paints to remove compounds potentially interfering with experimental processes. So we created a focus library of more than 30,000 elements that were processed for docking studies with human IDO1 crystal structure. We, among generated solutions, we selected the most energetically stable poses, those with Gleiges score inferior than minus 7.5 kcal per mole. Then these that were in the next step were typified and clustered by k-means algorithm, ensuring in this way structural diversity. 82 clusters were generated, where the compound with the best G-score of each cluster was selected for purchasing, but only 59 compounds were available. Before performing biophysical assays, we check the quality of our protein, our recombinant protein, produced by Giotto Biotech. Although only a small amount of the protein displayed heme, its prosthetic group inside the binding pocket, enzyme had a stable conformation over the time, and it proved to be able to convert L-Tryptophan in kynurenine, so it was active. At this point, we could start to analyze binding affinity, binding activity of virtual hits by biophysical assays. In the first screening, we performed NMR water-Loxy experiments to evaluate which fragments bound either one among purchased molecules. In particular, NMR water-Loxy is a biophysical technique based on magnetization transfer from bulk water to ligand-protein complex and finally to ligand. Each compound was analyzed twice, in a first experiment alone in solution and in the second one in presence of the protein, but in the same experimental conditions. We considered as binders compounds that in presence of the protein generated a spectrum with opposite sign or with a peak intensity difference measured than 90% compared to the first experiment. As a result, we obtained that 22 molecules were binders, giving a hit rate of 37%. In the next step, NMR hits were tested in MST experiments. MST, microscale thermophoresis, is a biophysical technique based on thermophoresis. Thermophoresis is a directed movement of particles in a temperature microgradient, depending on molecular properties such as size, charge, and hydration shell. Hence, each event, like a binding event, changing one of these parameters induces a variation in thermophoretic behavior that can be monitored in a titration experiment labeling target protein. For this reason, we labeled our recombinant enzyme with red fluorescent dye, according to nanotemper protocol, to perform MST screening. As in NMR assays, we use NLG919 analog as positive control because it is a potent IDO1 inhibitor actually in clinical trials. Thanks to KD values obtained, we could calculate BEI, Binding Efficiency Index, which links potency, expressed as PKD, with size of each fragment tested. In the next slides, we show you the structures of molecules tested in MST screening, where 14 compounds proved to be able to bind either one with different potencies. Here, we reported the 3 most potent binders identified that in the next step were characterized in their physicochemical properties to have a preliminary idea about their pharmacokinetic behavior in vivo. pKa and log-P results suggested that all compounds could display a good balance between solubility and passive permeability in vivo. Then we evaluated compound activity in cells. Before measuring their potency as a kynurenine fold change, and then as IC50 value. But in this case, we measured only the IC50 value only for the most potent inhibitor identified in the previously experiment, such as BIS-210. We obtained an IC50 value around 15 micromolar, which was in agreement with MSTKD achieved previously, around 18 micromolar. At the end, we performed delayed type hypersensitivity test to understand what was the final effect of our best compound, BIS-210, in vivo. As we can see in the graph, we can note that BIS-210 can stop the inflammatory reaction induced by immunogenic peptide administered to female mice. But this effect, this tolerogenic response, seems to be not dependent on IDO1, because this effect seemed to be more significant in mice treated with IDO1 knockout dendritic cells than in mice treated with wild-type dendritic cells. So these results suggested that this tolerogenic effect of BIS-210 depends on the interaction with other targets besides IDO1. Okay. Now,
So thank you, Alice. So on the basis of these disappointing results, we decided to go back and to investigate the biological relevant space sampled by our fragments. So we ran a principal component analysis using MolQ-D fingerprints on our internal database of more than 2,000 IDO1 inhibitors reported in patents and literature, and introducing also our fragments. Navigating the dimension of this space, we identified one compound, BIS-237, that actually was occupying a remote and unmined region of the chemical space that is located far away from the other molecules. Now, attracted by these observations, we further analyzed this compound. We observed that the molecules show a striking structural similarities with the known IDO1 inhibitor NLG-919. But still, it is devoid of any functional groups that would allow these molecules to directly interact with the heme cofactor into the enzyme. Now, attracted by these observations, we performed an analog-based screening around BIS-237, and we purchased and synthesized 20 analogs that were then tested in microscale thermophoresis experiments using the protocol that Alice was showing you before. Now, here we can see that these molecules are indeed able to further explore the unmanned region around the fragment 237, and here these 20 compounds are represented by the orange dots. Now, among tested compounds in particular, there was one, this 351, they show a very good potency in terms of binding activity. The association constant is 1.9 micromolar, so actually the same range of the analog NLG-919, and with an identical binding efficiency index of 90. So, attracted by these observations, we further tested these molecules in cellular assays, and we surprisingly found that these molecules is much more weaker in terms of inhibition potency of the enzyme with respect to the NLG919 analog. Indeed, the second compound has an EC value in cell that is in the nanomolar range, whereas these other molecules, this 351, has an EC50 in the micromolar range. Nevertheless, the molecules is still able to selectively inhibit IDO1 over TDO. Another interesting observation was that when we tested this molecule in a biochemical assay, in the case of NLG919, we were still able to detect an inhibition of the enzymatic activities, whereas our new molecule, this 351, was completely inactive. So attracted by this observation, we then tested the molecules in the immunization and skin test assay. Here we can see that indeed the molecule is still able to induce IDO1-dependent tolerogenic phenotype, thereby suggesting that there is a ligand-specific effect on the enzyme, and in particular that these molecules may indeed stimulate the signaling activity rather than inhibit the catalytic activity of the protein. Now, to further investigate this aspect, we analyzed how to design a signaling activity assay to better capture this effect. Now, as we have seen before, the enzyme has this eating motif localized at the level of the small domain that upon phosphorylation are able to recruit SHIP phosphatase and then induce long-term immunotolerance. So the first assay that we did was a phosphatase assay. In this assay, we use co-immunoprecipitation of SHIP IDO1 complex from cells treated with this 351 and from cells without any treatment. And we saw that for the co-immunoprecipitations from the cells treated with this 351, there was a higher phosphatase activity with respect to the untreated cells, indicating that in this co-immunoprecipitation, we have a large amount of SHIP phosphatase recruited on IDO1 structures. Furthermore, we also assessed the gene expression ability of these molecules in those cells. And we saw that treating these cells with 30 micromolar of our compound along a time of 16 hours, we were able to observe the expressions of genes which encode for proteins involved in immunocheckpoint modulator functions and in the long-term immunotolerance. So, finally, this molecule seems indeed able to modulate the signaling activity of IDO1 rather than inhibit its catalytic activities. And this would represent the first type of compounds that is able to do this kind of activity against this enzyme. So in summary, the overall hit rate of our fragment-based approach that was about 28% is higher than those reported in literature in high-throughput screening campaign of drug-like compounds for IDO1 inhibitors, proving the superiority of using a fragment-based approach or other approach in this kind of screening campaigns. Moreover, the use of fragment-based campaign has allowed to disclose one fragment, this Q10, with a high binding efficiency that is active as IDO1 inhibitors in cancer cells and can represent an interesting hit compound for further optimizations to develop novel inhibitors of these enzymes. We also were able to disclose one fragment, this 237, that was found to occupy an unmined region of the chemical space around IDO inhibitors, suggesting the high efficiency of fragment-based approach to sample remote areas of the chemical space. Now, charting the chemical space around this 237 has enabled us to discover this 351 that is a very potent binder of the enzyme with a high binding efficiency index that is actually the first ligand that is able to modulate the signaling activity of either one. So let me conclude acknowledging people that have worked with me and Alicia in this project, in particular the research group of Ursula Grohmann at the Department of Experimental Medicine of the University of Perugia, the group of Francesca Cantini at CERM of the University of Florence, and other people of my group that have provided their contribution to this research project indeed. So it's time for questions.
Thank you, Antonio and Alice, for your excellent presentations. Before we move on to our live question and answer session, I would like to invite you to provide us with some feedback on today's webinar. In a moment, 3 polls will appear on your screen asking you to rate the webinar. If you could take a moment to provide your feedback, it would be much appreciated. We now have the pleasure to introduce our question and answer session, where today's speakers will answer all of your questions. Don't forget, you can still submit your questions using the Ask a Question section situated below the player. Let's take a look at the first question. Have you already profiled signaling pathway responses in unbiased and quantitative manner? It would be interesting to see real global data showing changes in non-catalytic signaling activity and to then contrast with 1, Epacadostat mechanism of action and or 2, degree of IDO1 function modulation. We have some related experience and very relevant technology that might be of interest. Antonio, would you like to answer this one?
So thank you for these very interesting questions. We are indeed working on that, and we are actually optimizing a pharmacological assay that can provide us with a quantitative pharmacological parameter to characterize these compounds. And we are also comparing these activity values with those for the actual drug candidates, including Epacadostat, that are in clinical trials. Of course, any collaborations in this framework is very well welcome. So please, who of you is interested in establishing this kind of collaboration can email me and we can discuss in the next days how to establish this kind of collaborations.
Thank you. The next question is, which cells did you use for this kind of assay?
We use a cancer cell line that has P1HTR. I will pass now the word to Alice, that was responsible of the technical supervisions of the cells, so that she can answer to that question.
Hello. Hello?
Hello, so the question asked, which cells did you use for this kind of assay?
Sorry, can you repeat, please?
Yeah, of course. The question asked, which cells did you use for this kind of assay?
Okay, we use P1HTR cells, mouse mastocytoma P815 cells. Exactly.
Thank you. The next question asks, how does this interact with ACE inhibitors and beta blockers?
Sorry, excuse me. Can you repeat?
Of course. How does this interact with ACE inhibitors and beta blockers?
Sorry, but I cannot hear you very well. Can you repeat, please? Sorry.
Yeah, no worries. So the question asks, how does this interact with ACE inhibitors and beta blockers?
Sorry, but sorry.
Sorry, can you repeat, please, the question?
Of course. How does this interact with ACE inhibitors and beta blockers?
Okay. Now, this is part, actually, of a drug repurposing strategies that we are running. Indeed, the fragments that we have discovered show some striking structural similarities with this class of drugs, and actually we are actively studying these molecules as modulator of the enzyme.
Thank you for your answer. The next question asks, were your MST experiments performed with IDO1 in the ferric or ferrous form? And have you tried both and compared the dissociation constants?
Okay, what we have seen is that microscale thermophoresis, in our case, works very well. So we were really satisfied of the results obtained by microscale thermophoresis. In our screen approach, as we have seen, we used also NMR water-lopsy, but this type of biophysical techniques was used only on a qualitative basis to actually have the opportunity of comparing the MST results with the MR results. Again, in principle, we could have used only MST, but since this was the first time that we start using this kind of technology, we deem of usefulness integrating MST with another biophysical techniques in which our colleagues from Florence have much more expertise than my group, actually.
Thank you for your answer. The next question asks, did you analyze your inhibitor concerning AHR interaction?
This is another thing that we are doing, actually. So far, we have analyzed our inhibitor for HR transcription abilities using cellular assays. What we are trying to do is to develop also a biophysical assays for AHR. In terms of HR transcription activities, no one of these fragments is able to activate the receptor.
Thank you. Do you think that biophysical methods are sensitive enough to detect the binding of the fragment protein target? The size of the fragment is extremely small.
Yes, the size of fragment is very small, but biophysical methods such as MST or even surface plasma resonance are so sensitive that also minimal variation in their baseline is sufficient to reveal a change in studied system. It is indeed broadly reported in literature. They can very well detect weak interactions like those forming between proteins and fragments that show affinity usually more than 100 micromolar.
Thank you. How do you think in silico approaches will complement in vitro approaches in the future?
Computational approach will have in the near future more and more importance because on the one side that will allow an easier interpretation on in vitro results by providing access to big data repository in the web. So we will be able to interpret the new results, the new knowledge that we generate with those that is actually stored in database. Another aspect in which computational approach will help will be in assessing also how the conformations of proteins are induced by different ligands and how we can link these different conformations to different signaling activity of our target proteins.
Thank you for your answer. The next question asks, can you go over the technical advantages of using fragment molecules with a biophysical methodology such as MST?
As I already shown in the slide before, MST is a biophysical technique that is extremely sensitive. It can detect binding in a range of potencies from sub-nanomolar to low millimolar range of activities. So also wake interactions like those between fragments and proteins. So it will be the perfect choice for applying in drug discovery project a fragment-based approach. Now, in terms of the advantages of this approach, as I actually show you with these case studies, with fragments, we are able to efficiently sample even remote regions of the biological relevant chemical space. So we indeed can identify small chemical probes that are able to interact in narrow, shallow pockets of the enzyme or protein target and produce different signaling activities for the proteins.
Thank you. What type of binding buffer did you use for MST experiments?
I will pass Alicia for this to address these questions.
Okay.
Hi,
So,
Alicia.
hi.
So the question asks, what type of binding buffer did you use for MST experiments?
We used MST buffer that consists of trees and sodium chloride and magnesium chloride at pH 7.4. So, with DMSO, sorry, with DMSO, 2% of DMSO, and DTT, 2 millimolar of DTT.
Thank you for your answer. So the next question asks, is there a differential thermodynamic properties between ligands for acting at catalyzing activity versus ligands acting on signaling activity?
Okay. So at this moment we cannot. No. Okay. At this moment we cannot answer to this question because we have not enough information to answer.
Okay, that's fine. We'll move on to the next question. Why wasn't the biophysical method chosen for primary screening?
Sorry, what is the primary method for the screening? It's correct?
The question asks, why wasn't the biophysical method chosen for primary screening?
We choose NMR and then MST has the secondary screening because at the beginning we have a lot of molecules, a lot of compounds. So to reduce time and costs, we prefer computational technique to screen a very large library of molecules.
Thank you for your answer. The next question asks, how does MST compare to other technologies, such as NMR for FBDD?
Sorry, can you repeat, please?
Yeah, of course. How does MST compare to other technologies, such as NMR for FBDD?
Okay, MST presents a lot of advantages compared to NMR because we can use a very small amount of the protein and that allow to reduce also the number of false positive due to aggregation that can compare when we use a big amount of compound and a big amount of protein.
Thank you for your answer. The next question asks, what is the risk of detecting false positive or false negative at early stages with your approach?
Okay, in our approach, this risk is very reduced because we use the paint filtered during the virtual screening, and we choose fragments with high solubility, with log P lower than 3, that reduces the risk of aggregation. And finally, we, thanks to MSP, that can reveal quickly and the presence of aggregator precipitate, limiting the number of false positives.
Thank you for your answer. Unfortunately, that's all we have time for today. Any unanswered questions will be responded to via email. Please join me in thanking Antonio Masciarulo and Alicia Coletti for their time today. On behalf of Drug Target Review and Nanotemper Technologies, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.